The warm little pond hypothesis is much more recent than these other hypotheses, comes from 2017. And basically, what we're looking at is that meteorites are feeding some of these basic monomers, and they're ending up in these areas that have bodies of water where part of the year it's dry, part of the year we're getting liquid here. And basically, through uh, these cycles of dry and wet, we get these changing conditions. Um, and it allows these molecules to break down and then recombine. And so we can get these larger and larger macromolecules through this process. Now, I will admit I'm not super familiar with this hypothesis, but I want to throw it out there because it is something that's new that scientists are looking at for the origins of living things and these more complex molecules. Okay. Going from these polymers, right, we can chain these amino acids together. We can chain sugars together. We can build phospholipids. But that doesn't mean we've got living things. And as this says, we're never going to know for sure how the first cells came to be because we can't go back and look. But we can investigate this step by step to figure out potential pathways in which this could have occurred. Things that we know about cells. All living cells carry out metabolic reactions. They use energy in some way. They're enclosed within a plasma membrane, which is a phospholipid bilayer, and they can replicate themselves. So they can duplicate themselves to make copies. Cells have a genome of DNA that enzymes transcribe into RNA, and ribosomes translate that RNA into proteins. So we've got this central dogma of DNA to RNA to proteins. So we've got studies that support the hypothesis that cells arose from a stepwise process that began with inorganic materials. So we started with these non-living things. Bit by bit, we built something that looked a little bit more like life. And then we got something that looked even more like a living thing until we get to this step where we would now consider this thing living. Okay. And so we go from our inorganic molecules, which can build themselves in Earth space, organic monomers, okay, or amino acids and so on, can build themselves in aquatic environments on Earth. Okay. We see this happening. Polymers. Okay. So now we're building these larger molecules. This is where things get a little bit more complicated. Okay. And so early metabolism, something would have built these. Somehow we're getting these larger ones to build, self assemble as vesicles, become the first genome. So we'll talk about these things. We've got protocells in an RNA world. We're going to talk about the RNA world hypothesis. We're going to talk about protocells and how they form. Again, subject to selection. So we talked about natural selection. It was in play here as these non-living things were becoming living things. And then we get our DNA-based cells that we see in all living things today. So looking at the origin of the genome, this is our RNA world hypothesis. And what we believe is that we had an RNA system of inheritance before we got a DNA system. And so RNA would have been storing genetic information and then also acting like an enzyme, carrying out processes as proteins do now. So we've got these RNAs that function as ribozymes, okay, function as enzymes to carry out all of these processes, essentially making more of themselves, and then also passing on the genetic information in this way. And so I encourage you to watch these two videos because they've got animations and they're going to be explained much better than I will be able to do in the minute that I'm going to spend on this. But basically, what we're looking at is we get these various nucleotides. And in some situation, billions of years ago, they were able to self-assemble into RNAs, right? You get these random strands of RNA ribonucleotides, right? Our uracils, our cytosines, our guanines, and our adenines just kind of randomly bond together. And then they're able to function because of the shape that they take on. And they function as these ribozymes, and some of these are able to replicate themselves. And so, through the process of selection, these molecules that are better able to replicate themselves find themselves more and more. And then these other ribozymes that are able to pick up more nucleotides more quickly do better than their predecessors. And so we see this selective process where we are getting ribozymes, these RNA molecules that are better and better at passing on their own information, building more of themselves, and so on. Okay? And so that's the basics of the RNA world hypothesis. Now, looking at the plasma membrane, this is where we get this covering around these organisms, right? Eventually organisms. And this concentrates molecules and allows reactions to happen. So when we looked at the bubble model, that's a very basic membrane. When we look at creating a plasma membrane, it's much more efficient than the natural environment is to cause reactions to happen. So we're looking at protocells here that would have had organic molecules that would have formed as predecessors to living things. Okay, so we get these basic membranes created that are able to concentrate molecules and speed up those reactions. So we see small organic molecules can react with minerals and seawater to form vesicles with a bilayer membrane. Okay, so we actually observe in nature vesicles will form on their own. And if those vesicles just happen to have these RNA molecules in them, we've got something that's really starting to look like life. And so here's some protocells that are produced in the lab, right? We've got these membranous structures with molecules inside, and it's not super difficult to create these. This here is a TED Talk, and this is a scientist that looks a lot at protocells, and he talks here about the line that we draw between something being living and something being non-living. And in his work with protocells, he's really blurring this line, and it's quite interesting. So I encourage you to watch this video on protocells as well. So if we look at bacteria and archaea, these are our most our simplest of cells that exist, let's say it that way. Not as complex as our eukaryotic cells that we as humans have. Okay, so these first organisms 3 to 4 billion years ago would have been anaerobic. They didn't require oxygen, would have used carbon dioxide as their source of carbon to build more molecules. Because if you remember way back, carbon is a very versatile element, and it's the basis of all of our macromolecules. Okay. We see early fossil cells similar in size and structure to modern archaea and bacteria. So they're pretty small and they're unicellular. The first photosynthetic cells were bacteria that used a cyclic pathway. So they would have used CO2 and produced CO2. So it would have been a little while before we really get the photosynthesis as we know it producing oxygen. Looking at the Proterozoic era, we now got these cyanobacteria that produced oxygen through this photosynthesis using carbon dioxide, which would have changed the world entirely. And so basically what they're doing is they're creating all of this oxygen, changes the atmosphere. And because of this oxygen, we're not going to get the same formation of those molecules that we did in Earth's early history. And so we've got evidence of these early things through these stromatolites. They're basically mats that are created as these biofilms build one on top of the other. Okay, so as soon as we got this oxygen, we were done creating new mechanisms. And that's the life that we have, and that's the life that we see now, although different through the process of natural selection.